Good evening, everyone, and welcome to Science Today. We're here to interrupt your program since we're bringing you guys an urgent breaking news of a growing global threat, and it is about antibiotic resistance, which is putting billions of lives at risk.
ABR is routinely called the silent pandemic because of the way it lurks, relatively unnoticed and under-discussed, while wreaking havoc and claiming lives. Over the next 25 years, an estimated of 39 million people are expected to die from drug-resistant pathogens alone. That's over 5 times the number of deaths from COVID-19 in the last 4 years. Kind of crazy, right? And yet, for all the devastation, antibiotic resistance is largely absent from public consciousness.
Overuse of antibiotics is accelerating the antibiotic resistance among pathogenic microbes, which has become a growing public health challenge worldwide. Higher resistance means more severe infections, more complications, longer hospital stays, and unfortunately increased mortality rates. This puts an enormous strain on healthcare systems around the globe.
So, you guys might be wondering what an antibiotic is. Antibiotics are medicines that fight infections that are caused by bacteria inside our human body or inside animals as well. These medicines will kill the bacteria or slow down the growth of bacteria. However, if the bacteria in our body develop and defeat the drug that has been given to our body, we call it antibiotic resistance, ABR.
In addition, antibiotic resistance is when bacteria evolve to evade the effect of antibiotic through multiple different mechanisms.
So how do antibiotics work? I think our viewers would love to know. Okay, so antibiotics have 5 main mechanisms. The first one is inhibiting cell wall synthesis. The second one is inhibiting protein synthesis. The third one is inhibiting nucleic acid synthesis. The fourth one is metabolic pathways, and the last one is inhibiting cell membrane functions.
For the first method, we must understand that microbes have peptidoglycan cell walls. By inhibiting cell wall synthesis, the cell is vulnerable to osmotic pressure and autolysis. This doesn't work for animal cells as they don't have cell walls. For the second method, the antibiotics target the 30 or 50 subunit of ribosomes to inhibit protein synthesis, inhibiting the ribosome's function. Next, for the third method, the antibiotics inhibit the synthesis of DNA or RNA, respectively, usually by targeting the enzymes that assemble DNA or RNA nucleotides.
For the fourth method, antibiotics act as anti-metabolites. They do this by affecting bacterial growth, inhibiting it. Lastly, antibiotics can inhibit cell membrane function. For example, some antibiotics do this by acting on the lipopolysaccharide membrane portion of gram-negative bacteria, destroying it and thus disrupting cellular functions.
Thanks for explaining. From the many ways antibiotic works, people assume that it can solve everything with harmful microorganisms. Sam, can you explain why is it not possible?
Sure, this brings us to why we're making this news report in the first place. Microorganisms have developed something called antibiotic resistance. Let's look at how resistance occurs. So now let's look at how resistance occurs. So resistance can happen due to number 1, natural reasons, number 2, selective pressure, number 3, inaccurate diagnosis, number 4, self-medication, number 5, poor hospital environment, number 6, availability of few new antibiotics. Antibiotic resistance occurs naturally over time. This happens via genetic mutations and genetic material transfer. Genetic mutations occur spontaneously and can be accumulated. Mutations can happen in enzymes, amino acid sequencing, cell wall, cell structure, control genes, or chromosomal structure. Furthermore, genetic material transfer happens when it is transferred by a different genus or species of microorganism. In bacteria, this may be spread by antibiotic-resistant genes carried in plasmids or mobile genetic elements known as MGE.
Next is selective pressure. Some microorganisms may survive after treatment with antibiotics. Thus, survivors will multiply and take over the population as the dominant form. Inaccurate diagnosis is another cause. Professionals may misdiagnose symptoms and recommend a more widespread treatment where a more specific antibiotic is needed, or inappropriately prescribe antibiotics if there is uncertainty whether the illness is being caused by a bacteria or virus.
Adding on to those explanations, self-medication can contribute to antibiotic resistance because individuals may take incorrect doses of antibiotics. Inadequate doses may result in survivals. Overusing antibiotics or using them for conditions that don't require them can increase the selective pressure on bacteria, encouraging the development of resistant strains. Furthermore, poor hospital environments increase the likelihood of spreading infections. Lastly, we have the problem of few new antibiotics being available. Due to lack of knowledge and technical challenges, such as funding in other sectors being favored, the development of new treatment has slowed. This makes old treatments with proven effectiveness being overused, with new treatments only reserved for more serious cases.
To summarize, bacteria develop resistance in 4 main ways. Firstly, intrinsic resistance, where bacteria naturally lack structures targeted by antibiotics like a cell wall, making them naturally resistant. Secondly, acquired resistance, where bacteria gain new resistance through mutation. Thirdly, genetic change, where DNA mutations result in changed bacterial components and receptors, make antibiotics ineffective. Furthermore, genetic material transfer can occur between bacteria. Lastly, gene transfer, where bacteria exchange resistant genes through transformation by naked DNA incorporation, or transduction through phagocytosis, or conjugation through direct contact.
When antibiotic resistance becomes ineffective, the consequences are severe. The core issue with antibiotic resistance is that infections could be very difficult or even impossible to be treated. This increases the risk of severe illness, the spread of diseases, disability, or even death. The reality is stark. Common illnesses that were once easy to manage can become life-threatening. Medical procedures like surgery, case sections, and cancer chemotherapy also become riskier due to higher chance of untreatable infections.
And the impact doesn't stop there. Patients with drug-resistant infections often need longer hospital stays due to treatment delays and failed therapies. This puts a strain on hospitals, increases resource utilization, and raises healthcare costs. It also limits treatment options, interrupts regular hospital activity, and can increase morbidity and mortality rates.
These are serious challenges, but let's look at some real-life examples of antibiotic resistance on how it has already affected global health.
One prominent example of the impact of antibiotic resistance is the overuse of antibiotics during the COVID-19 pandemic. In 2020, over 29,400 people died from infections acquired in healthcare settings. Many COVID-19 patients were prescribed antibiotics unnecessarily, even though antibiotics do not work against viral infections. A study from a Chinese infectious disease center found that 71% of hospitalized COVID patients were given antibiotics, even though most did not have bacterial infection. Data also showed that children who were treated with antibiotics for COVID-19 had an increased risk of severe outcomes. This just emphasizes how dangerous ABR or antibiotic resistance can be.
Absolutely, and this isn't related to COVID-19. Tuberculosis TB is another major contributor to antibiotic resistance. Multi-drug resistant TB, or MDR-TB, is caused by bacteria that don't respond to the two most effective first-line TB drugs, isoniazid and rifampicin. MDR-TB is treatable with second-line drugs, but these are often more expensive, less effective, and more toxic. In addition, resistance to second-line drugs can also develop, making treatment even more challenging.
That's right. Patients with MDR-TB who don't respond to second-line treatment face limited options. This situation has turned MDR-TB into a global public health crisis. Shockingly, only about 2 in 5 people with drug-resistant TB access treatment in 2022.
So what we see is a domino effect. Antibiotic resistance not only endangers individual patients, but also put a lot of pressure on the healthcare systems worldwide. And that is why it's vital to recognize and address ABR right now. The stakes are high. Without action, the problem will only grow.
Now, let's take a closer look at groundbreaking developments in synthetic drugs, antibiotic innovation, and vaccines. Each bring new hope in our battle against antibiotics resistance.
On March 12, researchers made a major breakthrough by designing a synthetic drug that effectively shuts down protein production in bacteria. This new antibiotic showed significant success against drug-resistant bacterial strains in mice, although it hasn't yet been tested in humans. If successful in human trials, this drug could provide a new tool in treating resistant infections. This team, led by Dr. Andrew Myers at Harvard University, working with Dr. Yuri Polikanov from the University of Illinois at Chicago, has been exploring how to modify existing antibiotics to combat resistance. Their focus is on nincosamides, an older class of antibiotics derived from natural sources. With the National Institute of Health funding, they synthesized a new compound called cresomycin. Through X-ray crystallography, they analyzed cresomycin's ability to overcome the two most common resistance mechanisms that render other antibiotics ineffective. The findings were revealing. They discovered subtle structural adjustments that allow cresomycin to bind tightly to bacterial ribosomes, making it highly effective against resistant strains.
In other exciting developments, research published this October highlights the potential of vaccines in reducing ABR. Unlike antibiotics, vaccines don't just treat infections, they also prevent them. By preventing infections in the first place, vaccines reduce the need for antibiotics, thereby limiting opportunities for bacteria to develop resistance. Vaccines play a unique role by decreasing the selective pressure that allows resistant strains to thrive over non-resistant ones. They prevent disease, regardless of the pathogen's susceptibility to antibiotics, meaning resistance spreads slowly, if at all. And because vaccines target multiple antigens, often a key to the pathogen's ability to infect, resistance is even less likely to emerge.
So there are 6 ways to reduce the risk of antibiotic resistance. Firstly, take antibiotics as prescribed and never share antibiotics with anyone. Next, we should dispose leftover medications properly by returning it back to a pharmacy or communities that have not paid back programs to protect the environment.
Also, practicing good hygiene, like washing hands regularly, can also prevent infections and reduce the need for antibiotics.
Let me give more examples of ways to reduce the risk of antibiotic resistance. We should get ourselves vaccinated as vaccine can prevent infection that lead to using or avoiding the use of antibiotics. Additionally, supporting sustainable agriculture by choosing products from farms that use antibiotics responsibly and build relationship with local farmers for better meat sourcings.
And last but not least is to eat more plant-based protein foods, such as legumes, tofu, and tempeh. And also, lowering meat consumptions. Therefore, we're also using less antibiotics in animal farming. Further, it also helps to prevent resistance.
Thanks for tuning in to Science Today. We hope you have learned a valuable lesson. Please don't forget to like, share, and subscribe to continue spreading the science knowledge. See you next time.